let us look at the production of the amino acid lysine and threonine. So first coming to the microbial production of L-lysine. L-lysine is an essential amino acid which is present at a very low concentration in plant proteins. The pathway for lysine biosynthesis is shown here. We can see that the lysine biosynthesis pathway is a very complex pathway and it is starting from glucose. The glucose is going to glycolysis, going through glycolysis, it is forming phosphoenol pyruvate, it is being converted into pyruvate, and then this pyruvate is entering the citric acid cycle or the Krebs cycle, and it is being converted into oxaloacetate. Okay, so whatever oxaloacetate is formed, it is going into the pathway for the amino acid biosynthesis. So oxaloacetate is converted into aspartic acid, and then you can see here that with the help of aspartate kinase, that is the first step in the biosynthesis, aspartate kinase is converting aspartic acid, and the, the four different amino acids are formed here. When you're going down, okay, when you're going down, finally you're getting lysine, but in the same pathway, you also get other three amino acids, that is methionine, threonine, and isoleucine. Okay, so aspartate kinase is involved in the production of all the four amino acids. Then we have homoserine. These are the important enzymes, rate limiting enzymes. So we have a homoserine dehydrogenase, this particular enzyme. So from aspartate semialdehyde, what this particular enzyme does, it is converting aspartate semialdehyde into methionine, threonine, and isoleucine via different steps. So this particular enzyme should be very low if we want aspartate semialdehyde to go to the lysine biosynthetic pathway. Then we have dihydrodepicolinate synthase. This particular enzyme, what this does is, when homoserine dehydrogenase is taking it towards the production of methionine, threonine, and isoleucine, dihydrodepicolinate synthase is taking aspartate semialdehyde to the lysine biosynthetic pathway. Then finally, two other rate limiting enzymes are dehydrogenase variant and succinate variant of enzymes. Okay, so these two enzymes catalyzes the conversion of piperidine 2,6-dicarboxylate into DL-diaminopymylate, which is a precursor for lysine. And the difference between these two enzymes is the dehydrogenase variant is a one-step reaction for the conversion, and the succinylate variant converts it into the precursor in 4 steps. And the regulation of lysine biosynthesis. Okay, so for these are the regulatory process. Aspartate kinase, this particular enzyme is involved in the biosynthesis of all the 4 amino acids that has been shown in the previous slide. So, and all the 4 amino acids, if they are overproduced, this aspartate kinase can be inhibited by feedback inhibition. So if so much of lysine is produced, feedback inhibition go, aspartate kinase comes down, so no more lysine is produced. But we don't want this. So by genetic manipulation, it has been possible to create mutant of aspartate kinase that are insensitive to the feedback regulation, and hence more and more of the amino acid can be produced. The next enzyme, dihydrodepicolinate synthase. This is the enzyme, this is a key enzyme that is taking the pathway to the lysine pathway. So this has to be overexpressed in case lysine production has to be increased. Then we have the succinylate and dehydrogenase variant that is producing the precursor for lysine. So both these enzymes are also have to be overexpressed for lysine biosynthesis. And finally, DL-diaminopymylate that is involved in the formation of L-lysine from the precursor. These are also very important enzyme, and the regulation of this enzyme, the overexpression or overproduction of this enzyme will also increase L-lysine production. Okay, the microorganism used for L-lysine production, microbial synthesis in the fermenter are Corynebacterium glutamicum, Brevibacterium flavum, and Brevibacterium lactofermentum. And strain improvement have been done on these organisms to produce more, to make it more productive, okay, for the overproduction of lysine. So let us see what are the strain improvement that have been done. Mutant selection have been done, mutant selection, induced mutation and mutant selection had been done. So mutant organism was selected such that they are resistant to the lysine anti-metabolite beta amino ethyl L-lysine. So these resistant lysine antimetabolite resistant organisms are shown to have higher productivity for the lysine amino acid. The mutant strain with altered enzyme aspartokinase was selected so that it is not regulated by the end product inhibition. So there is no inhibition, so the pathway is keeping on continuing. Such mutants were also selected. The strain with decreased homoserine dehydrogenase activity, the enzyme taking it to the different pathway, the biosynthesis of the other three amino acids that have been selected so that all whatever is there, it is going to produce lysine. And then the strain with reduced citrate in this activity was also selected, and these reduces the occurrence of citric acid cycle and it has shown to increase the L-lysine production. So these are the different strain improvement that have been done on Corynebacterium glutamicum and Brevibacterium flavum, such that the productivity of the amino acid lysine is increased. So for lysine production, the carbon source can be sugar, beet or cane molasses. It can be starch hydrolysate. It can be sugar, acetate, and sometimes ethanol, alkenes, etc. And the nitrogen source can be ammonium salt and gaseous ammonia. Protein hydrolysate are also added into the medium so that it is supplying certain amino acids, that is methionine, homoserine and threonine. Why do we add these amino acids? These are also involved in this very same pathway like lysine metabolism. So if these amino acids are there already present, that means it will feedback inhibition will happen and these amino acids will not be produced anymore. So whatever is there, it is going to the lysine production. Whatever steps, it is going to the lysine production, and these amino acids are no further synthesized. Okay, so a little amount of these is added, and the protein hydrolysate, apart from giving the amino acid, it is also supplying the growth factors like biotin. So under optimal condition, the yield of lysine is 40 to 50 gram per 100 gram of carbon. Now looking at the downstream process, release and recovery, lysine is released out of the cell wall of the bacterial cell with an efficient of an effective carrier protein present on the cell membrane. So the cell membrane of these bacteria have a carrier protein that can transport lysine outside the cell. So this whatever lysine was produced, it is coming out of the cell. So now we, in order to produce a 50% pure L-lysine solution, after the process is completed, the biomass, the cell biomass can be separated from the solution, and then whatever solution you are having, you can evaporate it to reduce the water content, filtrate it to remove impurities. So this way you can get a 50% pure lysine. And to get 98 to 99% pure lysine, you'll have to go to ion exchange. You'll have to perform ion exchange chromatography, selects the amino acid lysine, elute lysine in the pure form, then evaporate off whatever water content is there, and finally crystallize it. The next one is threonine. Threonine also follows the very same pathway, but it deviates from aspartic acid. So the threonine production is 5-step process from aspartic acid, and the regulatory steps are aspartate kinase. Okay, the first enzyme, and homoserine dehydrogenase. These are the important enzymes involved, and these two enzymes is regulated by feedback inhibition by the product. So we can see that going down, threonine is produced, and from threonine isoleucine is produced. So we make sure that after the production of threonine, it is not converted into isoleucine. Whatever enzymes are there for isoleucine production is not there. So whatever threonine has been produced, it can be used. It is not further converted, and L-lysine and methionine is also not produced. So the organism used are E. coli and Corynebacterium glutamicum. Strain improvement have been done. Mutant with high level expression of THR ABC gene have been selected. This particular gene codes for 3 polypeptide, aspartate kinase, homoserine dehydrogenase, and threonine synthase. All the 3 enzymes are involved in the threonine metabolism pathway, not metabolism, threonine biosynthetic pathway that we have seen earlier. Then strain with minimal L-isoleucine production is selected so that whatever threonine you have, it stays. Then carbon source that are used as glucose, sucrose and yeast extract. Nitrogen source is ammonia, and the efficiency of production is 60%. Looking at the downstream processing, heating is done to coagulate the cell, And after the cells are coagulated and removed, filtration is done so that all the impurities are removed and water content is reduced by evaporation, and finally the amino acid is crystallized. So this is all about the microbial production of threonine and lysine.